and senior scientist at Children's Hospital of Oakland Research Institute, Nutrition and Metabolism Center in Oakland, California, where he is interested primarily in the prevention of cancer and other degenerative diseases of aging. Dr. Ames was awarded the National Medal of Science for changing the direction of basic and applied research on mutation, cancer, and aging. He established that many cancer-causing chemicals are also mutagens, that is, they cause mutations in cells, and he devised a simple, inexpensive test for environmental and natural mutagens, commonly called the Ames test. It has been widely used in research institutes, industry, and regulatory agencies around the world to screen for environmental carcinogens and mutagens and to analyze the mechanisms involved in metabolic activation of carcinogens. It has had a major influence in weeding out mutagenic chemicals before they are introduced into commerce. He has also identified the causes and effects of oxidative DNA damage and translated these findings into intelligible public policy recommendations on diet and cancer risk for the American people. Specifically, he concluded that degenerative diseases of aging, such as cancer, cardiovascular disease, cataracts, and brain dysfunction, are in good part due to oxidative damage. Dietary antioxidants such as vitamin C and E and carotenoids play a major role in minimizing this damage, he argues. During his career, Dr. Ames has tried to dispel many of the myths about the causes of cancer, chief among them that trace chemicals in the environment, such as pesticide residues on food, are a significant cause of cancer. The main causes of cancer, he argues, are lifestyle factors, ranging from poor diet to smoking and lack of exercise. A native of New York City, Ames obtained his bachelor's degree in chemistry from Cornell University and his PhD in biochemistry from the California Institute of Technology. Dr. Ames is a member of the National Academy of Sciences and was on the Commission on Life Sciences. He was formerly on the board of directors of the National Cancer Institute, also known as the National Cancer Advisory Board. He was the recipient of the most prestigious award for cancer research, the General Motors Cancer Research Foundation Prize in 1983, the highest award in environmental achievement, the Tyler Prize in 1985, the Gold Medal Award of the American Institute of Chemists in 1991, and the Glenn Foundation Award of the Gerontological Society of America in 1992. In 1997, he was awarded the prestigious Japan Prize. Dr. Ames has been elected to the Royal Swedish Academy of Sciences, the Japan Cancer Association, and the Academy of Toxicological Sciences. He's published more than 500 scientific articles. I would like to, I forgot one thing, I wanted to thank our own Bill Grant very much for making the initial contact with Dr. Ames, who then agreed to be our speaker. I would also like to acknowledge the help of Dr. Ames' assistant, Teresa Klask, who was very helpful in working with me in making arrangements and providing information that we used in our newsletter, Smart Life Forum newsletter this month. So without further ado, I'm delighted to present Dr. Bruce Ames.
Okay. Should I not? I don't need this, right? I'll turn it off and put it here. Okay. The introduction reminded me, Lyndon Johnson once was being introduced to give a speech in Texas, and he got a very flowery introduction. And when he got up, he said, "My father would have really liked that introduction, and my mother would have believed it." So So I'd like to talk, my interest is disease prevention. I was interested in prevention of DNA damage and then preventing mutagens from damaging human DNA, and that got me into cancer and preventing cancer. And now I'm interested in aging and delaying aging and trying to delay the degenerative disease of aging. And a lot of it has to do with nutrition. And so what I'd like to do is start off talking about our experiments on aging and how I think we're making some progress. And then in delaying aging, but then if you'd like to accelerate your aging, which about half the country is doing, I'll tell you how to do that. Yeah. So let's see, do we have a laser pointer around somewhere? No, okay.
Advance, push you forward and first near.
Okay, yeah, those two, yeah. So to make energy in the body, what you're doing is you're eating fuel, and the fuel is fat and carbohydrate and some protein, and you're burning that fuel in your mitochondria. Mitochondria, there are about 500 or so mitochondria in every cell, and these are the power plants of the cell. And what the mitochondria do is they burn fat and carbohydrate, and burning means pulling electrons from the fat and carbohydrate and combining it with oxygen. And so you add 4 electrons to oxygen, you're home safe to water. And that process pumps protons across the mitochondrial membrane, so you get a charge across the membrane. And so it's like a rechargeable battery. And that charge enables one of the complexes in the mitochondrial membrane to make ATP, which is the high energy molecule of the cell. And the ATP powers your muscles, it powers your brain, and powers all your biochemistry. And you're making kilos of ATP in your body every day. And that's all made in the mitochondria. But with age, what's happening is you get more 1 electrons addition. So if you're supposed to add 4 electrons to the oxygen to make water, but if you add them one at a time, you get superoxide and hydrogen peroxide and hydroxyl radical. And they're oxidants that can start oxidizing the mitochondria and the DNA and other parts of the cell. And with age, you put out more and more oxidants. So the mitochondria decaying with age, and more the degenerative disease of aging, Alzheimer's and Parkinson's, are being linked to mitochondrial decay. And so we've become experts in how to try and delay the mitochondrial decay. So as I say, I was interested in DNA damage. So what happens when your DNA gets damaged? You get a lesion in the DNA. These are the 2 strands of DNA. Normally, you know, they're wound around each other in a double helix. But for sake of clarity, I've put them flat. So what happens when you get a lesion in the DNA? Well, there are 2 kinds of repair systems. And these are enzymes cruising along the DNA looking for trouble. Well, one of them is nucleotide excision repair. And the principle here is this system doesn't care what the damage is. Anything that's a little bump that doesn't look like a normal base pair gets clipped out. Whether it can be synthetic, natural, doesn't matter. And so that's a good general system. And if you go out in the sunshine and make a thymine dimer, you can clip that out. Or if you're eating charcoal broiled meat and get a little benzpyrene, you get a benzpyrene on your DNA, it'll clip that out, or whatever. And then and then the other system is called base excision repair, and there are about a dozen enzymes that are looking for specific damages in DNA. If you deaminate a cytosine, you can get a uracil in DNA. And uracil isn't supposed to be in DNA, it's an RNA. And so there's an enzyme just cruising along the DNA looking for uracils. Now there, you don't really get a bump. You have normal, so it's a little different. And there's a specific enzyme looking for uracil in DNA. What it does is take the base off the sugar So you have just the ribose exposed, and then the phosphates are hooking up these riboses. Another enzyme comes in and makes a nick here, and then another enzyme copies this base and sticks in the right nucleotide and sews it up, and you've repaired it. It's like new. So, and there, as I say, there are a dozen enzymes, and half of them are for oxidized bases. So ever since oxygen came into the world, oxidation of DNA has been important. And these are very conservative enzymes. They're practically the same in E. coli bacteria and in people. So through all of evolution, these enzymes are recognizably the same. So that tells us nature thinks all those things is important. And as I say, about half of them are for oxidized bases, some for deaminated bases, a few for methylated bases. You can wave your arms and say why all these would be endogenous, we'd be getting this kind of damage. And so base excision repair. So one day I was walking along the Berkeley campus and I had an epiphany. Urine. People in Berkeley get high on all sorts of things, but I've been high on urine for a number of years. So, because what happens when there's a lesion in the DNA, this repair system cruises along and clips it out. And then what happens? It should go in the pee. So you're peeing out the history of your DNA damage every day. You guys are just throwing it away. So anyway, so here, what happens if there's a lesion, then all the other, everything gets clipped off, the sugars, the phosphates, and then that damaged deoxynucleoside, so the ribose plus the base, go into the urine. And here, you just get the base off the ribose, and the base goes in the urine. So we got interested in oxidation is coming from a byproduct of aging, and so we decided to see how much was going on. So the radiation biologists have done all the chemistry. They have reported there are about 100 minor products from oxidizing DNA and about 20 major products. So we started looking for the major products. Thymine glycol, adeoxyguanine, all these were known things. And we developed some methods and took a rat and put it on a defined diet and asked how many molecules does a rat pee out in a day. And then we got the number and how many cells in the rat, and et cetera. So our conclusion was that there are 100,000 oxidative hits to the DNA of every rat cell every day. And that number really shook us up. And we did every possible control we could think of, but we kept on coming out with about that number, no matter what base we looked at. So then I decided, wow, who wants to worry about a part per billion or something? This is what's really battering up your DNA. And we've stuck with that conclusion. So then we took normal rats on normal rat chow, which is a much better diet than your average teenager is getting, and asked, well, what's the steady state level of these oxidized bases in the DNA? Just a normal living rat. And it was about 24,000 in a young rat per cell in the DNA. And by the time the rat's old, it's about 67,000, which is pretty good repair systems. If you're getting 100,000 hits per day and you're repairing out it all the time. So we're doing very well. And rat does it and lives to 3 years, and we do it and live to 80 years. And so, does it really matter? But what's getting the brunt of the oxidation? The mitochondria, but that's where you're making the oxygen matter. And Earl Statman at NIH, who's a famous scientist, he's a man of very few words, but whenever he says something, you better listen, because he's a really deep thinker. And every 10 years, he changes his field and does something wonderful. Anyway, a while back, he got interested in oxygen radicals, and he started showing that if you look at protein, when you oxidize protein, you get carbonyl groups in protein, which they're not normally there, and you can measure them. And protein turns over. If you have an oxidized protein, then it gets turned over. But so he started looking at the steady state level of oxidized protein, and this is rats, and this is people. So it just goes up with age. And then progeria and Werner's syndrome, two diseases of premature aging, where these are cells in culture, we're aging much faster. And so your DNA is oxidizing, your protein's oxidizing, your lipids are oxidizing. We're measuring an oxidation product of fat going rancid. This is malondialdehyde. And so whatever tissue of the old rat you look at, compared to the young rat, there's more oxidation products. So we're all going rancid. My brain's going rancid, my everything else. Okay. So some of us faster than others. And so we got interested in all of this, and two terrific postdocs in my lab, Mark Shiginaga and Tori Hagen, and I wrote a review on oxidative damage in mitochondrial decay and aging. Now, that wasn't original with us. Denham Harmon had suggested that aging was just like radiation. You were oxidizing yourself, and Jaime Miguel. There were several people who were pioneers in this area, but we felt we contributed a few insights, and I became convinced that it was really true, that that's a major factor in aging. Now, aging's trade-offs. It's the theoreticians in aging don't think that we're programmed to go through a certain number of cell divisions and then die, because that doesn't fit with any evolutionary theory. And so they think it's wear and tear, basically, that life is all trade-offs. So, and I tend to agree with that. So anyway, we wrote a review on that, and then I decided, gee, we ought to work on that, because all the degenerative disease of aging have a big aging component. And Tori Hagen, who is a brilliant postdoc, had a lot of experience in mitochondria, and he started working out the methods. Now, the first thing he had to work out was that when you isolate mitochondria from old rats and compare them to mitochondria from young rats, and people have been doing that for many years, you're fooling yourself, because it turned out old rats, when you isolate the mitochondria, they're very fragile and very heterogeneous, and you lose half of them. So the enzymes end up in the cytoplasm, so you're breaking them up. Where young mitochondria are very stable and you get them out. So we first had to solve that problem, and he figured out a way to do that. So you want to look at a tissue that's not turning over all the time, and you want to look at single cells, and so white cells in the blood don't work very well because you're always making new ones. So you want to look at the brain or the heart or the muscle or the liver. So we ended up with the liver, and you can perfuse a rat with an enzyme which dissociates the cells of the liver, so you get individual hepatocytes. And they're not fragile. You get the same yield from young and old. And then we looked at mitochondrial function in the hepatocytes. And Hagen worked all this out. I was just cheering him on, really. And so this is what mitochondria in young rats look like. You see all the structure, the cristae. And here are old rats. They're all sort of fuzzy. Yeah. And so a little quick, sorry to throw all this biochemistry at you, but mitochondria then have an outer membrane that goes around like this, and then an inner membrane. So mitochondria are the most complicated organelle in the cell, and there are about 500 of them per cell. And they have their own little piece of DNA, too. And each, people think the mitochondria descendants, higher organisms, a yeast is a higher organism. It may not look like a higher organism, but bacteria are primitive organisms and yeast are higher organisms. And the reason is yeast have true nucleus and mitochondria, and bacteria don't. So people think the higher organisms, eukaryotes in technical terms, are descendants of one bacteria parasitizing another and then becoming slowly differentiating and ended up making the first higher organism. Anyway, so you have the inner membrane, the outer membrane, and then this is the inside of the mitochondria, and this is the cell out here. So there are 4 complexes that are called the electron transport chain. And lots of Nobel Prizes were awarded for the people who figured all this out. And so what they do, and the Krebs cycle, that's taking all the pyruvate that comes from your sugar degradation and running it around here to generate reducing power. And so basically what this passing these electrons do, till they all get to complex 4 here, is allowing you to pump protons across the mitochondrial membrane. So there's basically a charge across the membrane. So it's really like a battery. And that charge enables complex 5 here to make ATP. So ATP uses that high energy phosphate and goes to ADP, and now you regenerate the ATP by this rotary motor here, complex 5, and what powers that is protons coming across the membrane. So people really understand this in quite a bit of detail, and there's a fair amount of biochemistry going on in the mitochondria too. So with age, what's happening? Well, it's all degrading. So one is that the oxygen utilization in old mitochondria from old rats, I'll say old mitochondria, but they're really mitochondria from old rats, is much less. The membrane potential is much less, the potential across the membrane. The cardiolipin, which is a key lipid in the inner membrane of the mitochondria, which is essential for the accurate functioning of all these complexes, is down, and the oxygen radicals are way up. So, and the oxygen radicals, these oxidants come from not doing this process efficiently. So you have to get 4 electrons over to complex 4, and you add 4 electrons to oxygen to make water. So if complex 4 isn't working well, then you start leaking electrons around, and you end up with all these oxidants. So all those things are going wrong with the mitochondria. We developed methods for looking at all these things and just familiarize ourselves with mitochondria. And then in writing a review, we became interested in the work of some Italians from Bari, Italy, who published a paper saying if they fed carnitine, actually they used acetyl ester of carnitine, and carnitine's a transporter to get fatty acids into the mitochondria as fuel. So they fed it to old rats, and they said mitochondrial transcription got better. That is, that was making its RNA from its DNA better. And so he said, that looks interesting. Now there's a company in Italy that sells acetylcarnitine. It's sold over the counter as a pick-me-up. Now you'd think Italians are all picked up with espresso, but they seem to occasionally buy this stuff. And so people were a little suspicious about the paper because nobody ever heard of Bari, Italy. It's not on the tourist track. And this group was being supported by this company that sells this stuff. But it was a good paper, and my wife is Italian, so we go to Italy all the time, and I'd been to Bari, and I knew these people, and they're good people. And so I said, I really believe this stuff, let's try it. And we had all the methods, so we fed acetylcarnitine to old rats, and then tried everything. And everything got better except one thing. So the cardiolipin, which goes down with age, got better. The membrane potential, which goes down with age, I'll show you all the results in a minute. So you can see in old rats, the mitochondria the membrane potential is very heterogeneous, different, and also it's much lower than the young rats. And so the things that were going wrong are lower cardiolipin, lower membrane potential, lower oxygen utilization, increased oxidant leakage. So what we found was the acetyl, this is carnitine. It's only used in mitochondria. It's a normal biochemical, and its function is to esterify a fatty acid here, pump the thing into the mitochondria, take off the fatty acid, burn it, and pump out a carnitine to load up another fatty acid. And what you find in the blood mainly is acetylcarnitine. And this Italian company sold acetyl. They said it got through the blood-brain barrier a little better. We didn't know at that time about anything, but we just tried the acetylcarnitine, just put it in the drinking water of these rats. So we abbreviated ALCAR. So here, the young rats, old rats fed acetylcarnitine. And this shows young rats with and without acetylcarnitine, nothing happened. Doesn't do a thing to young rats, but old rats, it just moved this curve right over to here, make them much more like young rats. And then So then we looked, but what didn't get better were the oxygen radicals. These mitochondria, even though we fed acetylcarnitine, these old mitochondria were leaking out oxygen radicals. So we said, we need some special antioxidant. So we tried a few things that didn't work. And then Lester Packer's in our department, and he's been working on oxygen radicals for a long time. And he was touting lipoic acid, which vitamin E didn't work, and we tried ubiquinone. Anyway, we tried a number of things. And lipoic acid really isn't an antioxidant in the cell. It's a coenzyme for two mitochondrial enzymes. It's only used in the mitochondria. And the reduced form has two sulfhydryls, so it's a powerful antioxidant because these can get oxidized to the disulfide. So this is the oxidized form, and this is the reduced form. And it's attached to proteins in the cell. But a number of people had actually used it, tried it as an antioxidant, and it seemed to work pretty well in the cell as an antioxidant. And so we thought we'd try it. So we fed the old rats the oxidized form of lipoic acid, and lo and behold, the level of oxygen radicals came down to the level of a young animal. This shows young animal, we're measuring oxidants being produced, young and old. A lot more oxidants being produced, but old plus lipoic acid really was practically the level of a young animal. So the two together seemed to complement each other and solved all the problems that at least we had measured. So we fed the old rats the two together. And when our paper came out, I made the mistake of telling some reporter these old rats got up and did the Macarena. And I guess that was a good soundbite because it appeared in every news story all around the world. And then I got a very angry letter from a Scottish pharmacologist who chewed me out for about 3 pages that they really didn't get up and do the Macarena. It was very unscholarly. Scientists aren't supposed to do things like that. And he went on and on. He complained to the president of the university and everybody on down. It really upset him. But anyway, I wrote him back. It was a joke, but it didn't seem to mollify him. So this shows malondialdehyde, which is a product of lipid oxidation. And here are young rats, here are old rats. So they pour out a lot of malondialdehyde when they're old. So they're going rancid. And this shows plus lipoic helps, acetylcarnitine plus the two together. So you have the two together, it solves the problem of the malondialdehyde. And then we looked at all sorts of things on the whole rats. And the old rats are pretty lethargic, young rats are pretty frisky. And here are this is distance traveled at night. We have a video camera spying on the rats at night. They're nocturnal animals. And so young rats are moving around a lot. Old rats don't move around as much, but this peps them up. And then we looked at the immune system. The immune system goes down with age, and this makes it a little better. And then we looked at IQ tests. Now, IQ tests in rats are a pain in the butt, and we didn't know beans about it all, but you need to learn it, you learn it. So we went to all the experts we could find out how to do IQ tests in rats. So this is a standard one called the Morris water maze. Basically, it's a little pond with turbid water and a hidden platform. So you have visual cues all around, you have a platform hidden somewhere. And now rats can swim perfectly well, but they don't like to be swimming with nothing underneath them. And the first day you show them where the hidden platform is, and then you do successive trials on successive days. The visual cues are all the same. The young ones remember and they make a beeline for the hidden platform. The old ones wander all around the place. And this is more quantitative. This is young rats. This is distance traveled to find the hidden platform. As you can see, young rats really remember, and old rats wander around, plus acetylcarnitine, plus lipoic, and plus the two together. So then people said, well, you've done that, but it's just their eyesight going out. It's not their brains. So what do? I mean, that's a fair criticism. So then we figured, I knew a psychology professor at Cal, and Seth Roberts, and he had been at Harvard, and there's a famous Skinner box. Skinner was a famous psychologist at Harvard and developed this training system where you have a little box and you put the mouse or the rat in, and if the rat presses a pedal, it gets a food pellet. And so you make the rat hungry for a day and you put it in there, and then you train him to do various things related to this. And the one that Roberts had developed was you make a noise or shine a light, and 50 seconds later they have to press the pedal. 45 seconds, no deal. 55 seconds, no deal. So they have to remember a time interval, and they can do that perfectly well, or the young ones can. This shows sound and light are identical. It's the brains. So the green is the young ones, and the old ones are this burgundy color here, or whatever color that is. And in this case, acetylcarnitine is the yellow. It didn't do anything. Lipoic helped a little bit. That's this blue. I should get colors I can name. And then the two together were better than the sum of the two. So that's this. So I never brought it back to here, but it really made it better, and sound and light were the same. So we published all this, and then we made a monoclonal antibody to oxidize nucleic acid some years ago for 8-oxoguanine. And so we decided to look at the neurons in the brain. So this is young rats, they're old rats. You can see you're staining the neurons. And this is acetylcarnine lipoic and the two together. And if you, and then we looked at, so we assumed it was DNA, but in fact it turned out to be RNA. There's much more RNA in the cell than DNA, and the nucleus wasn't staining, and yet all the rest of the cell was staining, so we figured, maybe it's RNA. And then what you can do is before you do all this, you treat it with RNase or DNase, which is an enzyme that removes the RNA and removes the DNA, and they're very specific, and you can see the RNase removes it and the DNase didn't. So you're oxidizing all the RNA to 8-oxoguanosine. So now we were actually wrong on how we thought this was working. We just assumed that the lipoic acid, which is the oxidized form, is getting into the cells, getting reduced to dihydrolipoic, which homes in on the mitochondria. That's where it's used, and that's how it all was working. And that's what Packer thought and everybody thought. But in fact, it turned out it works in a different way. So there's glutathione is the main sulfhydryl antioxidant in the cell. Huge concentrations, 10 millimolar concentration of these things in the cell, which is a huge amount. So, and the sulfhydryl group is easily oxidized, so it's protecting the cell against oxidation. There's sulfhydryl groups in proteins, and the pharmacologists over the years had turned up something called the phase 2 enzymes. And these are 250 enzymes. 200 get turned on and 50 get turned off when you have a stress that oxidizes the cell or damages sulfhydryl groups in various ways. And they've worked out all the circuitry. Well, glutathione synthesis is under this control of these phase 2 enzymes. And when Torrey Hagen was in my lab, he realized that he measured glutathione, and it goes down with age. You give lipoic acid, it brings it right back up. So he said, hey, it's turning on the phase 2 enzymes. And then one of his students showed that's really true. So what happens is, so this is a paper showing all about the 200 or so phase 2 enzymes that get turned on in the key nerve pathway. And Jung Soo, who's now a postdoc in my lab, but was a graduate student with Torrey Hagen up in Linus Pauling Institute at Oregon State, he showed that this NRF2 declines with age, and glutathione synthesis is going down with age, and lipoic brings it all back. So not only are you acting as an antioxidant, but what you're doing is turning on all your antioxidant defenses. And now the pharmacologist Talley at Johns Hopkins had shown you eat garlic, it does that. You eat broccoli, it does that. The compounds in there that are weak oxidizing agents or weak alkylating agents that will turn on the system. The signal protein is a protein called KEAP1 that has 2 nearby sulfhydryl groups surrounded by basic groups, so it's very easily oxidized. And what it turns out, what lipoic acid is doing, it's the oxidized form, it's going like that. So it's basically just acting as a weak oxidizing agent and oxidizing these very easily oxidized sulfhydryl groups. And then the transcription factor, NRF2, NRF2, is bound to this KEAP1. And the minute these two sulfhydryls get oxidized, this pops off and goes into the nucleus and turns on all your defenses. Now, nature, our liver and our cells are just filled with defense systems. You irradiate a human cell, it induces antioxidant defenses because radiation is an oxidative mutagen. And you can irradiate a mouse, it now becomes more resistant to higher doses of radiation. Just the way cells are more resistant to killing once you induce this system. So we're just a bundle of defense systems, and they're all, if you use them, you make more of them. That's how we work, because we're living in a sea of toxic chemicals, because every plant has 100 toxic chemicals to kill off the predators. All plants can't run away, and they don't have claws. All plant evolution is chemical warfare. They're making all these nasty chemicals to kill off insects, to kill off animals, because they don't want to get eaten all the time. And so, but humans deal with all this stuff because we're have all these defense systems. You induce DNA repair, you induce phase 2 enzymes, you induce phase 1 enzymes. There must be 50 of these big systems that people have discovered by now that protect you against one thing or another. And they tend to be general systems. So you get an oxidizing agent into you, don't care whether it's synthetic or natural, you just deal with it by inducing your defenses. So I can never get very worried about a part per billion of pesticide. First of all, we wrote a paper saying that 10,000 times more pesticides you're eating from your veggies, just all the natural chemicals in plants. And, oh, well, if it's natural, we know how to deal with it. Well, we know how to deal with anything because that's how the body works. Anyway, that's another hour talk. But the really important thing doing your end is bad diets and smoking. So this is 231 enzymes get induced. This is part of the phase 2, and 31 repressed. After 24 hours and 6 hours, you've turned on 106, and by 7 days, you're down to here. So, and then we did some work. I won't go into this, it's a little technical, but on acetylcarnitine, how that's working. But we think we have a pretty good idea how these compounds are working, and they certainly synergize with each other. So, and even just looking at the mitochondria, we tried to quantify all that, and young rats, old rats, you have more undamaged mitochondria in young rats than in old rats, and then acetylcarnitine and lipoic make it better. So what about people? This says, more quarters, for God's sake, more quarters, senility, youth. Okay. So when we were doing all this, in a fit of enthusiasm, I called up my son in New York who's in computers and said, one of my students seems to be changing old rats into young rats. And there was silence, and my son said, that's all very well and good, but you let me know when you do the next step, when you change old people to young rats. So your children don't let you get away with anything, and my son has a very quick tongue. Yeah. So anyway, so obviously the next thing is humans, but the university said, hey, you really ought to patent all this stuff. And we said, nutraceuticals, you can buy them in the health stores, how can you patent any of that stuff? They said, don't worry, we'll put it in a patent. So they put in a patent for the combination. So there are hundreds of patents for lipoic acid. They sell it in Germany for diabetes as the neuropathy of diabetes, lipoic acid helps. And they sell acetylcarnitine in Italy as a pick-me-up. But if you put the two together, then you deal with our patent, or the university's patent. And so then, I wasn't thinking much, and then some businessman friend of mine who was just getting an MBA from Harvard who came to me and said, you know, I had to do a a business proposition, a business proposal for a course at Harvard, and I decided to do your stuff. And I got all excited about it. We should do a company. And I said, I'm too busy in the lab to do a company. He said, don't worry, I'll take care of all the detail. So anyway, he formed a company called Juvenon, licensed the patent from the university, and they sell the pills with a combination. So there's some 50% off tickets there if anybody wants it. You can look on the website. I put all my stock, founder stock in the company into a nonprofit foundation when it was started, and I don't get any money from the company. So I have no financial interest, though I'll have money to give away to science if the thing succeeds. Anyway, the company is doing well, and we're trying to use the best stuff possible and doing all of that. So, and one of the things the company's done is anytime somebody writes, they sold $5 million worth of pills last year with basically one employee. It's all web-based. And anytime we get a letter, and we've gotten over 5,000 letters, maybe it's to 6,000 by now, it goes into a searchable database because I want to see anytime someone complains about a side effect. And also, if somebody says, it grows hair on my head, and 10 people write us, I want to know about that so we can do a clinical trial. So anyway, that's what we're doing. There was one side effect that kept on coming up. 1 in 200 people or so complain about a rash. We got 25 letters about when they took the pill, they got a rash. But the other people don't. Okay. So we told them, stop taking it. But one of the guys who worked for the company, Ben Treadwell, how am I doing?
You have lots of time.
Okay, but I have lots of things to say. So I'll rattle on for hours, so I'd better, okay, I'll skip that. But anyway, we think we know what the cause of the rash is, and we've added one particular vitamin biotin to our capsule because we think that's going to stop the rash, and it seems to do that. But anyway. This says, you're 57 years old, I'd like to get that down a bit. So there have been 21 clinical trials on acetylcarnitine, no complaints about safety, and the meta-analysis says, yeah, it looks like it really works, but I'm not completely convinced. It's a small effect, so I wouldn't say you should write your mother about it, but anyway. And then lipoic acid is a little more convincing. There are about 6 clinical trials now, though I haven't made a new slide, and the meta-analysis says, yes, it really works for the neuropathy of diabetes. So now I'd like to switch gears. So that's all I want to say about lipoic acid, but I'm very optimistic. I think among these 5,000 letters, we got half a dozen letters from people, including one Nobel laureate I know who's taking the pill, saying I have high blood pressure, I measure my blood pressure every week. When I took Juvenon, my blood pressure went down. So my secretary, a Nobel laureate, and 4 other people told me that, or wrote letters in on that, and so we said, let's add that to a clinical trial we were doing, and in fact, it does lower blood pressure. So that was our first successful clinical trial, and we're going to do a cognition trial. We just did a dog trial. There's a company in Canada called CanCog. I don't know whether it's canine cognition or Canadian cognition, but anyway, there's a guy at the University of Toronto who set up a company on the side, and what they do is they take old beagle dogs. So you can have young beagle dogs, and they teach them all these tricks, and old beagle dogs can do the tricks, but they make many errors, and they just don't do it very well compared to young dogs. So if you want a compound tested, they test it on a dozen old beagle dogs. So they have 3 different tests, so Juvenon paid for these 3 tests, and the guy called us up all excitedly. It's the best thing we've ever tried in these old dogs. And so it improved 2 complex cognition tasks, but a simple task, just straight memory, it didn't improve. So we're happy with it, and the paper is sent off to the journal. And so we'll do a human cognition trial at some point. A lot of people have written us that their minds work better. But that's all anecdotal, and you really shouldn't believe it. Oh, so what this was is biochemistry. So I know you can't see this and I can't see this, but it's all the cycles and the metabolism. So people have worked all this out by now. So what do you need to keep your metabolism going? Well, you need fuel, you need fat and carbohydrate and protein, and then you need 40 micronutrients. The micronutrients are the vitamins and the minerals. So every living creature needs 15 more or less minerals. You can't make them, you have to get them from the outside. You need them in relatively small amounts, but you need them. And then you need 15 or so vitamins, and then essential fatty acids, essential amino acids. Anyway, those are called micronutrients. So what happens when you don't have enough micronutrients? It fouls up your metabolism. And I'll tell you why I think nature wants it, so when you don't have enough micronutrients, you batter up your DNA. Okay? So I got interested in this. So what, are we getting enough? We don't have scurvy around much anymore, or rickets and all the traditional diseases of deficiency. So everybody sort of forgot about it. But in fact, we're not getting enough. The RDA, they've changed all the names and nomenclature. It's a mess. But anyway, I'll briefly explain those parts that are in. So the RDA, you all know, the recommended dietary allowance, and that's the amount that will make the whole population have enough of this stuff. But 2 standard deviations below that, there's something called the EAR, and if you're below the EAR, that's kind of a measure of population inadequacy. Now, for iron, menstruating women aren't getting enough iron. 16% of the menstruating women don't have enough iron. And that's a disaster in terms of your biochemistry. Magnesium is a real disaster. The whole population, over half of the people in the United States aren't getting enough magnesium. And you look at teenagers, 80 or 90% of them. And obese are deficient in everything because they're eating junk food. And the elderly tend to be deficient in a lot of things. Zinc, 12% of the population. B6, elderly women, half of them. Folate, even with fortification of food, 16% of adult women are too low. E, 93%. C, 31%. So we're talking about a lot of people. So people aren't meeting the standards that the committees have picked. Does this matter? I think it's going to matter. So I got into this partly because I was interested in vitamin C and vitamin E because of antioxidants. But a fellow came to my lab on sabbatical, Jim McGregor, who's a very bright cytogeneticist. He's interested in chromosome breaks and things like that. And he had been working on a very easy method of looking at chromosome breaks in mice, where you just take red blood cells, which don't have any DNA, and stain them for DNA, and 1/2000 lights up. It has a little piece of DNA. And so when you make the reticulocyte, the precursor of the red blood cell, from a cell with a nucleus, when the nucleus gets extruded, you make the reticulocyte. But if there's a chromosome break, there's a little piece of DNA left behind. And you can just stain them and see them in mice. But so he was doing experiments with radiation. The more you radiate the mice, the more micronuclei you get, more chromosome breaks. And that was known. But then one day, all his control mice were full of chromosome breaks. What's going on? And he tracked it down. The technician hadn't put enough folic acid into the medium. And it turned out it was a deficiency of folic acid. He tracked it down, and anytime he made them folic acid deficient, bam, they broke their chromosome. So he had just come to my lab at that time, and I put my graduate student to figure out why, and we figured out why, we think. So when you don't have enough methylene tetrahydrofolate, these are 2 pools of folic acid. Folic acid's in the body is tetrahydrofolate. It's a reduced form, and it moves 1 carbon units around. So this adds a methyl group to uracil to make thymine. Now, uracil's in RNA and thymine's in DNA, so there's no uracil in DNA unless you've deaminated a cytosine. And so And we concluded that this was the mechanism. And if you're too low in this pool, what happens is homocysteine accumulates, and that gives you heart disease. And so, and you're always making more homocysteine. That's on the way to making methionine. Then you make S-adenosylmethionine, which is does all the body's methylation. And then you go back to homocysteine. You have to convert it here with vitamin B12 and methyl tetrahydrofolate. Anyway, there's, I could talk a lot about this, but I won't. But we thought we worked out the mechanism. But what about people? If you look in people, you don't see any micronuclei. And the reason is the human spleen, all the blood goes through the spleen. And after a few passages, you've cleaned out all the micronuclei, because the spleen takes out any red blood cell that's a little stiff. So, but now we've worked out a method for looking at fishing the reticulocytes out of blood, which are the newly formed red blood cells, with magnetic beads based on an antigen that's in the retic and not in the regular red blood cell. And that's only 1% of the red blood cells. And then if we look at that on the cell sorter, we can see the fluorescent ones and see the micronuclei. And we've shown it's higher in smokers and higher in thalassemia, where you release a lot of iron and et cetera. But I'm going to use that to get at what vitamin level we should really be having in people. Anyway. So the principle, radiation, the dangerous part of radiation is breaking chromosomes. And it also makes single mutations. But what people think is you get a cluster of electrons, or well, the radiation biologists think you get a cluster of electrons, and so you hit a base here and a base here near each other on opposite strands of the DNA. And you're always repairing these oxidized bases out from radiation, so you make a nick and you put in the right base and sew it up. But if you have two near each other, what happens is you start repairing them both out at the same time, and the chromosome falls apart. And that's what the radiation, so it's your own repair enzymes that are doing it in, because you have two nearby lesions. And you can repair double-strand breaks, but it's hard, and the radiation biologists think that's the most dangerous part of radiation. Well, by the time you have something like 1 million uracils in the newly synthesized strand and 70,000 or so oxidized lesions on the other strand just from living, you're getting one of these by chance, and you get a chromosome break. And that's what we see. So, so we think that that's the folic acid deficiency gives you chromosome breaks, and we can measure it. And so we looked in people. So McGregor had found one guy at Kaiser who didn't have a spleen, and they can live perfectly well without spleens. And he found a dozen people, and he talked them into giving him blood. And the normal range of chromosome breaks is down here. And he found this one guy with huge levels. So he followed him for about a year. And then he stumbled on this folic acid finding. And he looked to see what the level of folic acid was in this guy. And he had practically no folic acid. Folia is a Latin word for leaf, foliage. And where do you get folic acid? From spinach. So my mentor at Caltech, when I got my PhD, first isolated folic acid from 4 tons of spinach. So you don't eat your greens, you're not getting a lot of folic acid. So anyway, so he gave him reduced form of folic, and everything went down to normal, and then it started going up again, and he just put him on folic acid permanently. And then when he came to my lab, we went to Kaiser and got a lot more people and looked at people. This was the range of chromosome breaks we were getting, and then these people were off scale, and they turned out to have very low folic acid. And then we had a number of people who had moderate levels of chromosome breaks, and they turned out to be B12 deficient. And from the biochemistry, that should do the same thing. So, and then when we looked at what level of folate in the blood was causing this, I looked up who's at this level, and half the poor were at that level. I said, wow, this is important. Who cares about pesticides? We're breaking our chromosomes from not getting our folic acid. And there's a guy in Australia, Michael Fennick, who's also been showing folate deficiency and B12 deficiency breaks chromosomes in lymphocytes of men of Australia. So. So then next thing I looked at iron. So one of the professors in the nutrition department at Berkeley, Fernando Viteri, has been yelling and screaming for years about iron. Iron deficiency is the main micronutrient deficiency in the world because menstruating women are losing all that iron, and in the poor countries they're not eating a lot of meat, so they're not getting enough iron. And you can get it from veggies too, but they just were not getting enough. So there are 2 billion women and children in the world who are anemic or borderline anemic, and that's really bad for you in lots of ways. And the World Health Organization says, oh, we'll go in there, we'll give them iron, doesn't cost anything, it's rusty nails. So it's just a matter of will and getting it out to people. And, but again, it didn't work very efficiently. But the World Health Organization was giving so much iron, they were hurting them. And too much iron is bad for you. And so they never quite got that level right, and Viteri has been trying to get them to lower the level. But anyway, so one of his graduate students and one of my postdocs, we collaborated and we looked at the whole range of iron levels in rats. I always forget whether it's rats or mice. It was a while back. But anyway, I think it was rats. And so it turns out, and what we measured was all sorts of things about mitochondria. This had been one report that, that too much iron damages your mitochondria. So we were experts in mitochondria. We looked at mitochondrial oxidation of the mitochondrial DNA or mitochondrial efficiency and other things. And we've put it so up here is bad. So too much iron is bad for you, too little iron is very bad for you, and this is the range you should be in. Okay. So as, like most things, you don't want to overdo it. Mae West said too much of a good thing is wonderful, but I think she was thinking about sex, not vitamins. So you don't want to really overdo these things. And so anyway, for the metals, too much is toxic. Okay. So, and you start pouring oxidants out of the mitochondria when you have either too little iron or too much iron. So, and too little iron is associated with low birth weight babies, preterm babies, poor weight gain, et cetera, et cetera. And we've been writing a series of reviews. I have a brilliant older woman working in my lab, Joyce McCann, who used to be a postdoc with me 30 years ago and came back to the Bay Area and was looking for a job. And she's super smart and writes beautifully, so I hired her. And she's been doing a series of reviews on the brain growth spurt. While you're a fetus in the first 2 years of your life, you're making trillions of neural connections. And what happens if you're deficient in something? Well, your brain doesn't develop well. So there are whole huge literature on iron deficiency in rats, in mice, in people. And again, so she is a toxicologist, so she went all through this very critically and wrote a review, which is just going to be coming out, and it's irreversible. After a certain age, then it's irreversible. And and the poor are just eating this horrible diet. I think they're destroying their brains. And this shows DHA, which is the long omega-3 long-chain fatty acid, 30% of the fatty acid in your brain. And where do you get it? Deep sea fish, or you can get it, make it inefficiently from linolenic acid, which you can get from flaxseed and nuts and some things like that. But you should be eating a fair amount of fish. Well, the poor don't eat fish, or a lot of them. And then choline, and now we're doing one on vitamin D. So Bill Grant will be interested in that. Vitamin D, the whole northern tier of the United States tends to be somewhat deficient in vitamin D. But the people who are really deficient are dark-skinned people, because in the tropics, in Africa, southern India, New Guinea, racially completely different people, they have a lot of melanin in their skin. Why? Because it protects them against too much UV in the tropics. Otherwise, you get burnt and eaten by a lion or something. So there's a selection for dark skin in the tropics. And in Sweden, you need every little bit of sunshine you can get to make that vitamin D. You need a half hour or 20 minutes, Bill Grant can correct me, of sunshine a day to make the vitamin D you need for your metabolism. And vitamin D is really a hormone, and the brain is absolutely chock full of vitamin D receptors. It's doing something up there. And so we are putting together the case that really all dark-skinned people should be getting vitamin D because I think it's doing it in their brains. But that's very politically incorrect and controversial, but we'll see how it comes out. You start pouring out oxygen radicals. So I had a postdoc, Emily Ho, who is interested in zinc, and so she took human cells and culture, made them a little zinc deficient, and whammo, they start pouring out oxygen radicals, get massive DNA damage. You knock out P53, which is coordinating repair systems. You knock out APE, which is a repair enzyme for DNA that has a zinc in it, and so does P53. So you're doing all this nasty stuff. So, and this shows DNA damage by the COMET assay, and its oxidative damage. So one of my postdocs, Hania Tamna, worked out why. So it turns out that in the mitochondria, you're making heme. Heme, because it comes right off the Krebs cycle with succinyl-CoA and glycine. So heme is the red color when it has an iron in it. It's a chelating, it's a cage for an iron. And heme is in your hemoglobin, and you're making huge amounts of it. But it's also used in biochemistry quite frequently. So a lot of your biochemistry has heme in particular enzymes. And you're making all that heme in the mitochondria. And if you don't have enough iron, you don't make enough heme. You don't have enough zinc, which is involved in the second enzyme of heme biosynthesis. You don't make enough heme. You don't have enough biotin. You don't make enough heme. And what happens? Well, heme A, which is a fancy form of heme with a couple of groups on it. It's isoprenoid like cholesterol and a few other changes. And heme A turns out to be what goes out first. And where is that used? Only in complex 4 in the mitochondria. So there's an excess of complex 4, and the activity of complex 4 goes down. You can still make your ATP, but the reason there's an excess, it keeps oxygen radicals down. So the minute you start lowering complex 4, you start pouring oxygen radicals out into the cell. And that's just what happened. So, and now we're filling in this, so complex 4 is here, and it has an essential heme A to put the whole thing together, about 100 proteins in here. So there are about over 1,000 proteins that are made in the nucleus and shipped into the mitochondria with little barcodes on them, so they go to the right place. And then the mitochondrial DNA makes about 10 proteins or so. So this shows biotin deficiency in human cells in culture, and they just light up because they're full of oxidants. This is a fluorescent dye when it's oxidized, and it's not fluorescent when it's reduced. You put it in, and it oxidizes, and it glows. So, and biotin's used for the Krebs cycle and making things. And so, and what happens when you take human cells in culture and make them a little biotin deficient? They age sooner. So primary human cells go through a certain number of population doublings, and then they senesce. Well, they senesce earlier if you're a little biotin deficient. So we're slowly filling in this table, and these are all the micronutrients involved in heme synthesis, and we know that complex 4 goes out, and we've shown it for this one, this one, this one, this one, and this one's in the literature. This is in the literature, and these we've shown, and then oxidative stress we've shown for these, and DNA damage we've shown for these, and early senescence we've shown for these, and we've just sent the paper off on biotin, and so we're slowly filling this in. And so then I started thinking, well, folate deficiency gives you DNA damage, and all these things that give you DNA damage. Why the hell is nature doing it this way? And then it hit me that nature wants it this way. Think of the 15 metals. Every living creature requires 15 metals. Are they there in equal amounts in the world? No. What happens if a creature is in an area where magnesium is deficient or iron is deficient? Well, and by the time an organism starts starving for magnesium, what does nature care about? Survival. anything long-term gets cut out ruthlessly. So I think over all of evolution, you've adjusted binding constants for magnesium, so anything that's involved with DNA repair goes out first, and if it's important in your heart or your mitochondria or something that you really need, then you keep it there for the last thing, because you want survival so you can reproduce, and you don't care about anything long-term. So long-term, there's a trade-off between long-term survival and short-term survival. And so it makes sense, I have to prove it all, but I think it explains why we now must be up to close to a dozen different micronutrient deficiencies give you DNA damage. And then I looked in the literature, and there's really a lot out there on that. This shows magnesium deficiency, which I say practically everybody in the country is deficient in magnesium. It makes cells senesce early. And all sorts of nasty things happen to you when you don't have enough magnesium. Where do you get magnesium? It's in chlorophyll. Magnesium is in the center of the chlorophyll molecule. And so every time you eat spinach, you're getting magnesium. You might get a little E. coli occasionally, but they were the biggest organic food producers in the country, I guess. And so, you know, people say, organic food. But we're talking about parts per billion of the pesticide. And organic farmers use manure and things like that. The big ones probably don't, but the little ones do. So, and there are many more cases have been reported for E. coli from organic food than ordinary food. So if it tastes better and it's 50% more expensive and you want to spend the extra money, fine. But I don't think it's really helping your health. And what's good for the environment is to get more food out of less land. And you'd never get the yield of crops in the country if you didn't use pesticides. So
There are studies that show that you actually get higher micronutrient intensity in organic grown food.
I've seen some papers like that, but I'm not convinced, because you can grab this and that and do a study, but I wasn't convinced they had looked enough and done the right statistics. But I wouldn't say that depends on the, I sometimes buy organic food because it tastes better, but in general.
Why would it taste better?
Pardon?
Why would it taste better? Maybe it's more metal.
No, well, it tastes better because sometimes organic food tastes better. Sometimes I get non-organic food that tastes better. So anyway, I won't get into that. I can give you an hour talk on that sometime, but not now. Yeah. Anyway, this is another measure of senescence for magnesium deficiency. And this shows mitochondrial DNA, protein DNA crosslinks for magnesium deficiency. And in the literature, I found all kinds of deficiencies have been reported. To break chromosomes would be a risk factor for cancer, and we're finding more and more all the time. It's all buried in the literature, and nobody's taking it seriously. And we're experts in cancer prevention, and I've devoted my life to figuring out what's causing cancer and how to prevent it. And I must say, pesticides are very low on the priority scale and very hypothetical. There's really not much convincing evidence, and it's not anywhere in the same league with bad diets. We know obesity is linked to 40 different diseases, including cancer. So anyway, this is my triage theory about why all this is happening. And so I'll just say a few words about obesity. I'm a regular reader of The Economist, which is a wonderful magazine, and this was the cover once. And as we all know, there's a lot of obesity, and there are really 40 different diseases linked to obesity. So it's a public health disaster. So thin people are going to be paying the medical costs of fat people for years to come. So obviously, eating too much is one factor, but I think it's more complicated than that. If you look at what the 10 leading energy sources are in the United States, number one is regular soft drinks. 7% of the energy is coming for sugary soft drink. And you look at kids, they're drinking 6 of these things a day or something. Cake, sweet rolls, donuts, pastry. This is cumulative. Hamburgers, cheeseburgers, meatloaf, pizza, potato chips, corn chips, popcorn, rice, rolls, buns, English muffins, bagels, cheese, a cheese spread, beer, French fries, fried potato. Not a lot of greens on this list, and not a lot of vitamins and minerals either. So people are eating high-calorie, cheap, refined food, and they're not getting the vitamins and minerals. Now, 40% of the population takes multivitamins. I'll bet everybody in this room does, but it's not the poor. They tend not to do that. And they're the ones who really need it. Now, exercise is important too. So we all know you're supposed to be exercising. I stole this from some guy at NIH, Alan Spiegel. Yeah. So we all know we're supposed to be exercising. So it's the things your mom told you: eat your fruits and vegetables, and not too much meat, and eat some fish, and et cetera, and get a little exercise. But people aren't doing it. This. So visceral fat is the bad fat, because that's the fat that one of my colleagues is figuring out obesity. He's a former postdoc of mine who's independent now. And I think he's really going to figure it out, because he's doing wonderful experiments in rats. And anyway, but this visceral fat is just pouring out cytokines and doing all sorts of nasty things. So I stole this from some British website and modified it a bit, but anyway. So why not just take a vitamin pill as insurance? Well, the nutrition community hates the idea of vitamin pills, and Mark Twain was poking fun at vitamin pills, But so why should the nutrition community hate the idea of vitamin pills? Well, they say, we don't understand all the things in fruits and vegetables, these plant phenolics and all sorts of things. And if you tell the poor to take vitamin pills, then they'll never eat any fruits and vegetables. But they've been trying to get the poor to eat fruits and vegetables for 30 years with not much success. Why not tell people, take a multivitamin mineral as insurance, and then eat your fruits and vegetables, because you don't understand them. Anyway, so I think they're being very short-sighted, and I just got livid because I was at a conference at NIH, and the only thing they were interested in was randomized clinical trials. If you don't show that a multivitamin mineral pill works in a randomized clinical trial, you shouldn't recommend it. Now, the fact that some sizable percent of the country isn't getting the level they say, and that there are all sorts of epidemiology and biochemistry and theory and all this stuff, no, you shouldn't recommend it unless you do a randomized clinical trial. Well, there are a couple of successful randomized clinical trials, one in China and one in France. But if you're going to measure cancer, you have to wait 10 years and use huge populations, and nobody's going to do that kind of thing because it's enormously expensive. And so the few trials that have been done haven't been done very well, and so it's like you're going to wait forever. And so I think it's a very short-sighted policy, but anyway, so I've written a paper blasting that. So, anyway, to finish up, obviously, the more things, you need fiber too. If you want fiber, madame, I suggest you eat the menu. So you want fiber too, and you get that from your fruits and vegetables, and you need it for good gut health and all of that. And so, but anyway, life expectancy gets longer every year. This is, remember, we're at the turn of the century, 1900, people weren't even living to 50. And now women's life expectancy is just up to about 80, and men are not catching up, but not too far. And women have a much longer life expectancy than men. Some of that is genetics, but single men have an 8-year shorter life expectancy than married men. And both men and women know why. Single men self-destruct. They drink too much, and they smoke too much, and they don't eat any veggies. It's the wives that civilize them. So both men and women know that. Yeah. But anyway, I think it's going to get longer and longer. It increased, 25, 30 years over this century. It's going to increase even more next century, because we're learning what aging is all about. So I used to know the dozen people in the world who worked on aging. Now there are thousands of bright young kids working in the field. The field's ripe. We have all the tools. Money's sort of short, but I think we're going to learn about aging, and we're going to make a difference. So I'm an optimist. Anyway, we'll see how it goes. But you have to behave. You can't smoke and eat bad diets. Thank you.
Okay, can we take just
Yeah, a few questions. Yeah.
Okay. Yeah, by the way, before we take the questions, remember, Jim Carstad's got a videotape of this, so, you know, there's a lot of great stuff there. You guys make a note to buy the videotape next month when it's available. So, first question.
Yeah.
Yes, sir.
Yeah, we've had someone up here spoke about conflict between taking biotin and alpha lipoic acid at the same time, because chemically, for some reason, one destroys the other or cuts the benefit.
No.
What have you got to say about that?
Well, they both come off the same biosynthetic pathway. There's an octanoic acid, an 8-carbon fatty acid, that goes this way to make lipoic and this way to make biotin. And they're both needed by the body. And if you put more of one in, you can interfere somewhat with the other. So when Ben Treadwell, who works with Juvenon, learned about these rashes, and we were getting, as I say, one in a couple of hundred letters complained about a rash when they took it. We said, stop it. Well, he was on San Marcos, he lives on San Marcos Island, and he's the health guru on the island. And one lady said, gee, I really want to take the Juvenon, but when I took it, I got a rash. And he said, why don't you try an experiment? Wait till the rash all goes, and take a biotin pill every day. Maybe you're marginally deficient in biotin, and the Juvenon's pushing you over. And why don't you take a biotin pill and then take the Juvenon and see then do you get a rash? And she didn't. And he did this on another person, same result. And then we added biotin to the product, and since then we've gotten very few letters about rashes. So we think it's actually working, but we have to write a paper on this.
Yeah, by the way, I hope it's clear to everybody that Juvenon is a combination of acetyl-L-carnitine
and lipoic, and a little biotin, right. There, as I say, the 50% off tickets there if you want. Yeah.
What kind of doses are you talking about of the acetyl-L-carnitine and lipoic acid? And do you recommend alpha lipoic over R lipoic?
Okay, so R lipoic and alpha lipoic. Lipoic acid on the market has been all the racemic mixture, which means it's synthetic material that has the natural isomer and the unnatural isomer. When you do a chemical synthesis, sometimes you get both. And to get the pure natural product, you have to separate this out, which makes it cost a lot more. Plus, that would have been okay, but it turns out that this chemically synthesized stuff was a little more stable than the isolated R lipoic. So the R lipoic started polymerizing and doing nasty things. And so I was afraid to add the R till I was satisfied about the purity. So we, like everybody else on the market, was using the alpha lipoic, which is the synthetic mixture. But now, Geranova sells an R lipoic that's pretty good. Carlson's really good. And we asked some chemists to make crystalline salts with a lot of things. And you have to be careful what you're putting in, so you don't want to put in too much sodium, so because people are getting too much sodium anyway. Well, we did the magnesium salt, and it looks pretty good. And so we're eventually going to switch over to the R. But it's taking time, and I have to be satisfied about the purity and all of that. We're actually working with the Geranova people. Yeah.
The dosages?
The doses, you can get them on the Juvenon website, and they're the doses that we're still a little uncertain about the dose. We had to guess at what level had they done in clinical trials that didn't cause any trouble, and what level worked in the rats, and make a few guesses. So we're looking to get some measures that we can do directly in people to optimize things. But anyway, it's all on their website, so you can look up there. Yeah.
What is the advantage of Juvenon products over taking individual components?
Well, you can buy individual components in the health food store, but then no money's going to Juvenon to do clinical trials. So, but plus we're using the purest stuff we can and doing good quality control and figured out the biotin stuff. So that's how it is in the nutraceutical business. It's hard to defend and there are lots of rip-offs, so you don't want to spend all your money on lawyers. We want to spend the money on clinical trials. So all I can say is that it's going to a good cause. I'm not making any money out of it, and so hopefully, yeah.
Yeah, there have been numerous studies done that state that the vitamins don't really extend longevity that much compared to eating a good diet. You mentioned the diet. What food would you recommend to extend life and be healthier?
Well,
Broccoli's one, and what other vegetables?
that's a complicated thing. It's what your mom told you. Eat lots of fruits. The Mediterranean diet's good. The Japanese are living a long time. So you want to eat a good balanced diet, not too much meat, some fish, and lots of fruits and vegetables.
Any particular vegetables and fruits?
No, greens and nuts are good. And Seventh-day Adventists have half the cancer rate and live a couple of years longer. And there, I was at a epidemiologist meeting, and Malcolm Pike was explaining that the Seventh-day Adventists have half the cancer rate and live 2 years longer, and they don't smoke and they don't drink and they don't like caffeine and they go to church every week. And someone said, well, what do they die of? And he said, boredom. So, yeah.
I was wondering if you have looked at what triggers the decline in these cycles, what's actually causing the mitochondria to make its changes. And I'm wondering if there's some relationship to hormones because, in general, because I think what's happening is that as our hormones decline, we start to generate more of these illnesses of aging, which you see are showing up in the mitochondria. So could you
Yeah. Well, yeah, so nature, what nature really cares about is reproduction. And when you're old, does it matter much? I mean, you're not reproducing anymore. So, and for most of human history, a 40-year-old was a really old person. Life expectancy was relatively short. And so there's not, the selection has been for reproducing early and often, and there wasn't great, maybe the wise old woman in the tribe or the wise old man brought knowledge and it was somewhat useful to the group and they prospered a little better if some people lived long, but there wasn't strong selection for that. So, and your hormones, once your reproduction hormones change and all of that, then basically nature's forgotten about you. And so, I mean, that's a more waving your arms kind of explanation, but
is, what's the trigger that causes this decline?
well, it's just you're declining all the time. You're making extra oxygen radicals and you deal with them, but it doesn't pay nature to deal with them 100% because you have other things, reproduction and other things to worry about. So you're always getting damage and you repair it, but you never repair it 100%. And the lysosome, which eats up 10% of your mitochondria every day, keeps the bad mitochondria out, and that helps. But eventually the lysosomes get clogged up, and and there's cross-linked material that's hard to digest, and so life is complicated. But I think people are going to figure these things out and be able to push it back. But it's going to be a year here and a year there. You're not going to tomorrow have Methuselah. Yeah. Okay.
In the cell, normally without the lipoic acid, there's lots of oxidation going on.
Yeah.
And how come those oxidants don't turn on the secondary oxidation control mechanism?
Okay.
And lipoic acid, not a really strong oxidant,
Yeah.
can do it.
Okay. So lipoic is very small amounts in the cell. It's just used as a coenzyme. But we have antioxidant, vitamin C, vitamin E. We have antioxidant enzymes, superoxide dismutase and catalase and others. So we have layers and layers of defense and glutathione and all these things, but they're never just 100% perfect. So, and you can get damage to the DNA and then you're in trouble and other things. So the system just slowly goes down. It's like an old car slowly wears out and your engine starts putting up more black smoke and it's less efficient. Now, the body repairs things and you can repair your car, but eventually you junk your old car. Bye-bye. I'm almost 78, and so I'm going strong, but you never know.
But there's the protein you talked about, I can't remember the name of it, that when it's oxidized, then turns on a lot of genes that produce antioxidants.
Yeah.
Secondary process.
Yeah, this keep one nerve system.
Right.
Yeah.
And so, how come
It isn't turned on full blast.
lipoic acid is particularly good at oxidizing that protein?
Well,
There's so
but
many
it,
other-
as I say, garlic works too. So you can eat garlic all the time, be inducing it, or broccoli, and you choose your. That's maybe one of the reasons fruits and vegetables are good for you. But
That bird, I believe, did some research in developing a mechanism to deliver alpha-lipoic acid. Are you familiar with any of this? That bird called glucose.
yeah, he used to bug us all the time on the phone. I never knew what happened to him. But anyway, so I guess he has a company. I haven't followed that. Yeah. So it's you get into the aging business. There's a lot of hype out there and a lot of going far from the science. We're trying to keep our company close to the science. Yeah.
I was poking around in MD Consult today, and I noticed an article which said that there in certain sections of the population, the folate level was inversely related to risk of colon cancer.
Yeah, there was just a new, all the previous papers, there are about 5 different types of cancer that have been linked to low folate. Walt Willett has done some of this stuff and others. But there was just a new paper, which I have in my backpack somewhere here, but I haven't read, that says there's a kind of, at a low level, this seems to be protective. Now, folate's one thing that's a little funny about folic acid. As I say, my mentor at Caltech, Herschel Mitchell, first isolated folic acid from 4 tons of spinach. In the course of it, they oxidized the folic acid. Folic acid in the body is all reduced folate, which means it's fairly easily oxidized, but that's the coenzyme is reduced form. And now the oxidized folate works, and that's what's in vitamin pills, but people differ very much on how well they reduce it to the dihydrofolate and tetrahydrofolate. So that's the one vitamin I'm a little nervous about because it's not quite the same thing as the natural vitamin, and there seems to be some differences in the population. So it may be, anyway, I just assume that they used a reduced form of folate in the vitamin pill, but that there's some stability problems and some cost problems, but eventually I suspect they'll go to that.
You mentioned folinic acid there.
Yeah, that's a reduced folate.
A ratio of folinic to folate. They're now selling folic acid with different ratios.
Yeah.
Folinic and
Yeah.
folate.
Well, I think they should probably just use folinic or methyl tetrahydrofolate. But anyway, the committee, they'll work that out as papers come out and people figure all these things out. We're doing a little work in that area. Okay, I need to drive back to Berkeley. And so I'll.
Wonderful job. Thanks very much for coming. There's a ton of good stuff in there.